Good morning, students. Welcome back to my channel. So, in this class, we have covered about the basics of proto-oncogenes and oncogenes. What are making the proto-oncogenes converted into oncogenes? Now, you have seen the thumbnail. In this class, let's talk about what are the defects that happen during a DNA repair mechanism. Further, you should be aware of that DNA gets exposed to many number of damages daily. I can say the DNA damage is ubiquitous because every day DNA gets affected by some or the other thing. But we have got such a nice DNA repair mechanisms in our body, which gets immediately changed. Like it could be a chemical alteration, like that DNA particular damage could be a chemical alteration. Please remember that DNA damage is different from DNA mutation. So DNA damage is something which can be corrected, or it can be controlled, or it can be by some reason it have got repair mechanisms well, that this can be controlled and set back. But for a DNA mutation to happen, this cannot be controlled. It could be a nucleotide sequence change. So I told you in the last class, like point mutations or missense mutations or nonsense mutations, these are all the mutations which are permanent, which cannot be, you can't get back the DNA to a normal stage. So for that, first you have to know what is actually a DNA damage. So I told you DNA damage is ubiquitous. So you can't just pinpoint that it is because of this the DNA has got damaged. It's get exposed to many number of chemicals, or it gets exposed to many number of radiations and all. But still, the DNA repair mechanism is well that it keeps repairing all the time. So DNA damage is ubiquitous. DNA mutation is different from DNA damage. As I told you, DNA damage is a chemical alteration and can be connected, whereas the mutation which comes due to the change in the base pair sequence cannot be corrected. So it may lead to a DNA completely damaged, permanent damage in the DNA. Right. So now let's see why. So this comes the topic why the DNA repair mechanisms fail. So for that you have many things, like it could be intrinsic factors, it could be extrinsic factors, and also genomic instability, the basic requirement. So if you have gone through the short that I have given you, where it highlights the hallmarks of the cancer, what makes the main cell converted into a cancerous cell, one of the important hallmarks being the genomic instability. So what is genomic instability? There is no proper stability maintained in the genomic sequences. Like the gene is actually controlled by three maintained nucleotide sequences. So we have adenine, guanine, cytosine, and thymine. So based on the rearrangement of these three nucleotides only, you have your gene properly coming. Right, when you go across the translation process, you'll get through it. So when this gets disturbed, it could be due to the mutation, that could be due to the point mutation, where one nucleotide gets substituted with the other. If you have, for example, if you have a nucleotide which is coding for a valine amino acid, right? So if you are just removing that particular nucleotide and you are just substituting that with other nucleotide, that doesn't code for a valid, so it codes for some other proteins, or there could be no protein at all coded by that particular genetic code. So which leads to the a mutation in the DNA which cannot be controlled or repaired. So that is what you call genomic instability. So what are coming under genomic instability? Point mutations, missense mutations, and genomic translocations, right? These are all coming under. And you have intrinsic factors, and also you have the extrinsic factors. Let's all discuss in detail what are the mechanisms that fail when DNA is getting a mutation, right? So right. So these are the genomic instability, causes of genomic instability: point mutations, gene duplications. This in the last class I've covered you gene duplication. And also I also told about deletions and inversions, where the chromosome when there is a homologous chromosomes pairing up during a cell process or cell signaling, or during the cell cycling happens, there could be the exchange of chromosomal material, which could lead to either deletion of permanent deletion of a chromosomal part, or it could be a addition of your chromosomal part, so leading to a deletion and the inversion, also causing the genomic instability. So what are the and also the translocations I discussed in the last class. I will also attach that about proto-oncogenes and oncogenes and types of mutations. Please go through it. Okay, these are all the trinucleotide repeated expansion. These are all coming under the genomic instability. Now let's discuss about what are the intrinsic factors. So what is genetic predisposition? So some people or some Individuals might be having the threat of having a cancerous gene right from their birth. So at the time of birth itself, the gene that causes the cancer is present in the gene itself, like the predisposition right from their right from their birth itself, because of the genetic factors or genetic history of that particular family. That could be one of the important intrinsic factors for a cancer development. And aging, right, where as you keep aging, the repair mechanisms also would become so slow, right? The DNA repair mechanisms would not be so effective, and thus aging is also confined to a cancer development. Epigenetic changes, where environmental changes would also affect on the chromosome, leading to a cancer development. Now now let's see what are the extrinsic factors apart from this. Okay, the main extrinsic factors being the environmental changes, right? Viral infections, diet and lifestyle. So environmental carcinogens, so it could be, for example, if you're exposed to more and more UV radiation, right, it also leads to that. And if you're exposed to more and more smoke, so because of tobacco, right? So tobacco does contain many number of carcinogens, which have also covered in this class, I mean covered in the last I will also attach how the smoke really causes cancer in individuals. So if you want, you can go through that, what are the types of chemicals that are present in the tobacco smoke. And also secondhand smoke, whoever does take that smoke, that is what you call as second hand smoke. That second hand smoke is also one of the important carcinogens for the cancer. And you have the viral infections. I told you human papilloma virus is directly linked with the cervical cancer. That is why they are taking injection against that human papilloma virus by the time the girl is getting into her puberty. And you have your hepatitis B vaccines, hepatitis C vaccines, where the defect of that virus would also enter into a liver cancer. And diet and lifestyle, so high amount of the processed food and high amount of meat consumption, or high end amount of fried products. Okay, these are all and also having the sedentary lifestyle and doesn't have any exercise, and these are all the factors, and more obesity and sugar, having a diabetes. These are all the basic lifestyle changes in future may trigger into the cancer. Okay, these are the extrinsic factors that I want to talk about. Now let's see uh, what are the DNA repair pathways. So right, this is the important part, right? So base excision repair, double stranded break repair defects, and you have your nucleotide excision pathway. These are all the pathways how the DNA repairs when the DNA gets damaged to a DNA gets damaged in a cell, right? How it really you know does it. So it could be mismatch repair, or it could be a base excision repair, or double stranded break repair. So let's see what are the cancers that really happen. For example, if you take the nucleotide excision pathway, this nucleotide excision pathway is generally gets affected when there is over and over exposure of your UV radiation, right, leading to the skin cancers. Okay, and if you talk about the base excision pathway, so base excision pathway generally goes for a lung cancer. If any changes that happen in the repair mechanism gets defected, it leads to lung cancer, breast cancer, and colorectal cancer. If you have a mismatch repair defects, so mismatch repair generally happens during the replication process. If there is no proper replication process and the defects are observed in the replication process, then that repair defects would immediately lead to either colorectal cancer, or it could be endometrial cancer also, mismatch repair defects. Okay, and if you go for double standard break repair process, it generally leads to prostate cancer and lung cancer. Yeah, so these are the basic repair mechanisms where the defects happen. So if you want to know the further repair pathways, I will also attach the important slide in the next coming classes. If you have any doubt, please let me know regarding the repair mechanisms. Okay. Yeah, so these are the how the cancer cell is getting, how the normal cell is not able to repair the DNA, and thus leading to a cancer. These are the repairing pathways defects in it. Okay, hope you are clear with the concept of cancer, types of mutations, what are genes that are converting into proto-oncogenes and to oncogenes, and what are the factors that would make the genome unstable, and also you have covered in the shorts about the hallmarks of cancer, what are the basic hallmarks of cancer. And I thought of giving you a class about amino acids coding. Like, you have 20 different amino acids, right? So out of 20 different amino acids, what are the codons that code for the particular amino acid? When you have a translation process undergoing, what codon codes for amino acid? This could be a question in many exams, so I will also let you give you that in the coming classes. So if you have any doubt, please let me know in the comment box. Thank you and have a nice day. Please try to answer the questions that have been posted in the community tab for more information. Okay. So thank you and have a nice day.